welcome to the University of Birmingham. In these labs we study cancer research, but more specifically we are looking at angiogenesis. So angiogenesis, angio, which is from Greek meaning blood vessel, and genesis, which is from Latin meaning production of. So angiogenesis is the production of blood vessels. Now tumours can't really grow more than about 2 millimeters in size without having more blood vessels to provide them with oxygen and nutrients. We are trying to destroy these blood vessels. Kind of imagine it like if you are cutting off the supply lines to an enemy's fort. They can't build their fort if they don't have the supply to do it. So how do we do this? Well, everything in cells is controlled by genes. And if we can trick the vessels into turning off a gene that grows new blood vessels, then we can get some of the way into preventing these blood vessels from growing or destroying them. Cells are very good at protecting themselves from things that will damage the most important part of the cell. And the most important part is its DNA, and the DNA is where all of the information and its genetics are held. Imagine DNA like the hard drive on your computer. You don't want to do work on and save it onto your hard drive and then take your hard drive into work and transfer the work that way. You'd save it onto a pen drive, you take the pen drive to work, and that is how you transfer it. And this is how cells work. They copy their DNA onto a pen drive, which we call RNA, and that goes out into the cell and does all of the work and provides the information. Viruses can hijack this, and viruses have a type of DNA which is we call double-stranded RNA. And their job is to try and take this RNA, this double-stranded RNA of theirs, and put it into our genes. It's kind of like when you get someone else's shopping list and you add your items so that they go and buy the stuff for you. But cells realise that viruses do this, and what they do is they look at this code within the virus RNA and they sequence it, and they know exactly what this code is. They destroy it, but they also destroy every other sequence that's the same. Now we hijack this mechanism. We put into the cells a double-stranded RNA code that is the same as a gene that we are wanting to turn off. When we put this into the cell, the cell recognises this code, will sequence it and destroy all of the pen drives that are coming from the hard drive of the cell. So all of the RNA that's coming from the hard drive or the nucleus of our cell is destroyed by the cell as well, because it's the same sequence as that RNA double-stranded gene that we have put in it. So if we put in a sequence that is the same as a gene that controls the growth of tumour blood vessels, we can turn off this gene and so prevent the growth of new blood vessels. Now, we're developing tiny spheres called nanoparticles that we attach these RNA or molecules to deliver them to the cells. And this is where our research is really progressing. We're developing these tiny spheres and with the hope of targeting specifically and delivering huge amounts of this RNA so that we can turn off these genes for prolonged periods of time and hopefully stop and destroy new blood vessels from growing.